Namaste. In today's lecture, we continue our discussion on distance sampling. So, as we saw in our previous lecture, this is the distance sampling formula, as you can see on your screens. N hat is equal to Na by 2WL p hat. So, what essentially it is telling us is that n hat which is an estimate of number of animals in our area of interest and this figure n hat because it is an estimate, it is given by area into an estimate of the density. Now, area into the estimate of the density is given by the number of animals that we saw there as modified by the detection probability. So, it is n by p hat into 1 by a, and a is the total area of our samples. Now, in case the sample is strip plot, we have A is given by the number of samples into the length into twice the width. So, what we are referring to here is that here is our transect line and this is the half width W. So, we have half width W on the other side as well, and we want to calculate the area of this rectangle. So, this rectangle has a width of twice W and a length of L. So, the area of the rectangle is given by L into 2W. So, putting it back here, and K into L is also written as capital L. So, capital L is K into L, which is also the effort. So, effort is the total distance that we have moved. So, putting it all back there, we get n hat is equal to A into n by p hat into 1 by 2 k L W. So, we can also write it as n A by 2 k into L is given by capital L. So, you have 2 W capital L p hat, which is our formula for the distance sampling. So, for the estimate of the number of animals in our park or the area of interest. So, now distance sampling can be done in different flavors. So, as you can see on your screens now, so there are different flavors of distance sampling. One is that whether do you want a line transect or a point transect. Now, a line transect is what we discussed just now, in which we have moved a certain distance in a straight line. So, this is our line transect. Now, in place of a line transect, the other variation could be that we just stand at a point inside the forest, and then a point transect is generally used for birds. So, if you are standing at a point in the forest and then you have a number of trees around, so if there is any bird on a tree, then we are looking for the distance of the bird on the ground level. So, these are the distances as given by R1, R2 and so on. So, we can use these distances in the similar manner as we had used these W's. So, in place of W we can have R when we have a point transect. And rest of the calculations would be very similar. Another flavor, as you can see on the slides now, is perpendicular distance versus the radial distance. Now, what do we mean here is that when we are having a line transit, so this is the line on which we moved from A to B. Now, at every point when we see an animal somewhere, we want to know its distance from the line transit. So, if you remember, we had drawn a curve between distance from the line and the number of animals seen. Now, this distance from the line can be given by this straight line distance from the point of observation and the angle that the animal is subtending with the transit line, or it can be given as a perpendicular distance. Now, we know from our trigonometry that if this distance is, let us call it x, and if this portion is y. So, in that case we have y by x is equal to sin theta, or y can be given as x into sin theta, where theta is the angle that is subtended between by the animal and between the direction of the line transit. So, when we are putting our values in the computer, we can go either for the perpendicular distance directly, or we can give it in the form of a radial distance. So, you need r and you need sin theta in this case. Next, we have exact distance versus group distance. What this means is that suppose we saw a group of animals. So, we have 5 animals and we are moving on this line transit, and this is our point of observation O. So, when we are putting these distance values in the software, we can either go for the distance d1, which is for the first animal, then a distance d2 for the second animal, then a distance d3 for the third animal, distance d4 for the fourth animal, and a distance d5 for the fifth animal, and all their corresponding thetas. So, now this is one way of putting our distances in the software. Otherwise, we could go for one other thing. We, in place of putting all these different d1, d2, d3, d4 and d5, we can just assume the center of all of these animals. So, let us presume that it is this point. So, we can just put in a single distance d and a single angle theta, and then we can put in our software that there were 5 animals, n is equal to 5 at distance d and angle theta. So, when we say exact distances versus the group distance, this is what we mean: exact distances of each and every animal, or a group distance for the group. Now, a group distance also becomes important because in a number of cases there might be another animal here, say the sixth animal, which we could have missed out just because it is standing on the same line of sight as the animal number 3. So, the animal number 6 would get occluded by the third animal. So, we could miss this animal. So, it is always preferable to put distances as a group distance, so as to increase the level of precision in our calculations. Another way in which we use the concept of group distances or clusters is when we have our transit line from A to B, and when we are writing the distance of an animal or a group of animals, we can either write it as, say, d is equal to 31.3 meters, but in that case we are not completely sure whether this point is the exact center. Your exact center could be, say, this point. So, in some cases, in place of writing 31.3 meters, we could also group this distance as 30 to 40 meters. Now, in the case of distance sampling, while it is always preferable to have as exact a distance as possible, in some situations group distances become important, especially in situations when we are doing an aerial survey. Now, in an aerial survey you have an aircraft and on the wings we have put some struts. So, the struts are basically distance estimators. So, we can just say that this is say 50 meters, this is 100 meters, this is 150 meters, this is 200 meters and this is 250 meters. So, when we are observing a point at this particular height from the point of observation, and so this is our transect line A to B. We saw an animal here. So, the distance between the animal and the line of the transect D would be given by this figure of 50 meters. So, this is around 50 meters from the transect line. If an animal is found here, for instance, so we have only this reading that it is less than 50 meters, but we do not know exactly where this animal is lying, because our scale that is used on the aircraft wing is not that much precise. So, in that case our readings would be 0 to 50 meters and we saw an animal, then 50 to 100 meters, 100 to 150 meters and so on. So, we will just go on putting the tally marks as we see the animals. And this is another variant of putting a group distance. Another variation is whether we have a direct sighting or an indirect cue. Now, in the case of direct sighting, what we are doing is when we are walking on the transect line, we are actually observing an animal. So, in the case of large sized animals such as elephants, we can directly observe the elephant, but in some cases when it is difficult to sight the animal directly, we could even go for indirect cues. Now, what do we mean by indirect cues? So, suppose you are walking on a transect line and there is a tree nearby, and on this tree you saw the nest of a bird. So, this is a nest of bird. Now, if there is a nest of the bird, there should be a bird who has built this nest. So, because in most cases it is difficult to observe a bird directly, so we could go for these indirect cues. So, in the case of indirect cues, we would just observe the nest, we would locate its distance and bearing from the transect line, and then we would apply a correction factor. So, for instance, in the case of those birds which breed in pairs, we can just say that if we have seen a nest, then there must have been a male bird and a female bird. Now, this situation is only possible when we know of the characteristics of the birds. So, for instance, in the case of weaver birds, also called baya birds, the male has a tendency of building a number of nests, and all of those nests would be half woven. So, if you go to the forest, you would see nest hanging that is just half woven. So, it would look like this, when a complete nest looks something like this in a cross section. So, when you see a half woven nest, it means that the male bird has built that nest, the female bird came there, inspected the nest and did not find it suitable enough. So, actually they did not pair up for that particular nest. So, any of those half nests would only mean that there is a male bird who has built all of these nests, but we cannot say for sure that there is a pairing. But when we see a complete nest like this, then we can be completely sure that there is a male bird and a female bird that have built that nest and are residing there. So, these are known as indirect cues. Now, indirect cues could include the nests, or in some cases we can include pellets or dung. In some cases we could include even bird songs into our calculations. Now, pellets are important because in the case of some animals such as the elephant, an elephant is also termed as a mega herbivore. So, it spends close to around 10 to 14 hours every day just eating. This is because its digestive system is not efficient enough to process the large caloric requirements that the elephant has. So, essentially it would be eating a lot of leaves and grasses, and then it would be defecating them. So, when you are moving in a transit and you see that there is a line of dung trails, then you can be sure that it was one elephant. But then these dung trails could be of could be fresh dung trails, or these could be old dung trails, which can always be made out by looking at the texture of the dung. Also, things like footprints of elephants or tracks of other animals could be used as indirect cues. So, this is also another variant. Another flavor could be active detection on the field versus passive detection with camera traps. Now, in the case of active detection at the field, what we are doing is that we are moving on the transit line and we are observing the animal then and there. In the case of camera traps, we deploy camera devices such that whenever any animal moves in front of it, it will take a picture. So, this is known as passive detection, and we can utilize those pictures also as our data points in the transits. Now, the next concept is that of a detection function. The next concept is that of a detection function. Now, detection function, which is given by g of x, is a representation of the probability of detection of an animal at distance x from the transit line. So, essentially when we are having a transit line from A to B, and suppose there are 3 locations of animals. So, this is x1, x2 and x3. Now, the probability of detection of the animal at x1 would be different from the probability of detection of an animal at x3. Now, this is because when we are having an animal that is very far away from us, then in those situations it is very difficult to see that animal directly, and there is a very high probability that we are going to miss that animal. So, at distance x3 we will have the probability of detection, say it is p3 and here it is p1. So, we will have p1 is greater than p3 in most of the situations. There are exceptions, but in general we can say that if the animal is close to us, we can see it more clearly as compared to a situation in which the animal is far away from us. So, the probability of detection of an animal which is close to us is greater than the probability of detection of an animal that is far away from us. Now, what about the probability of a detection that is right in front of us, that is right there on the transit line? We can say that the probability of detection of this animal would be 1 or very much close to 1. Now, why are we moving into this concept of g of x or the probability of detection? Well, in the earlier class we had seen that we had this value of p, which is our detection probability, and it depends on a number of variables. So, it depends on the animal itself, it depends on the transit, it depends on where you have laid this transit, whether animals frequent this area or not. It depends on your mental state, it depends on the clothes that the observer is wearing, it depends on the food that he or she has eaten, it depends on whether whether the observer is making any sound or not, it depends on the perfume that is being worn by the observer, it depends on whether the observer is moving singly or is moving in groups. So, there are a number of factors on which this value p depends. Now, we wanted to make an estimate of p. Now, an estimate of p, just because it is an estimate, will depend on making some generalizations. So, one such way of deriving the value of p hat is g of x. Now, with this function probability of detection, we have said that if there is an animal that is right there on our transit line, we would say that the probability of detection is 1 at distance 0 from the transit line. So, we can write it as g of 0 is 1. For any other distances, we can write it as g of x. Now, when we say that g of 0 is 1, what we have done here is that in the case of our previous curve, we had distance versus number of animals detected. So, we had drawn all these bar graphs, and then to get the value of p, we had drawn curve that was moving smoothly through all of these bars. Now, in this situation, the top of the curve could be anywhere depending on situation to situation, but when we are doing when we are writing the function as g of x, we have said that g of 0 is 1. So, in that case, we can scale this curve upwards or downwards, so that g of 0 is 1. So, this is one mathematical simplification that we have done. So, the scaling is done in a manner that g of 0 is 1. In other words, the probability of detecting an animal at 0 distance from the transect line is 1, or that any animal on the transect line itself is always detected and is never missed. So, we draw a smooth curve over the frequency distribution and scale it to ensure that g at 0 is 1. So, this is the curve that we have gotten. Now, once we have received this curve, so the making of this curve is now simplified because we have these bars. We made a smooth curve and then we scaled it such that the top is at 1. Now, once we have this g x, how do we get the probability function, the value of p hat? Now, since p hat is the estimate of probability of detecting an animal, we can compute it for any width w by finding the area under the curve of g of x, which represents the probability of detecting all the animals present in a width of w, and dividing it by the area of the rectangle of height g 0, which represents the probability of detecting all the animals present in a width w when the detection probability is 100%, since g of 0 is 1. So, what we are saying here is that, let us remove this. Now, at any distance, say w, at any distance w, the number of animals that are detected is given by the area under this curve till a width of w. So, it is given basically by this area, and the total number of animals that were there is given by the area under the rectangle of width w. So, it is given by this area. Now, since we had written that p hat is the number of animals detected divided by the number of animals present, we can write it as the number of animals detected is the area under this with these blue hashes, which is given by an integral of g x dx varying from 0 to w, divided by the number of animals present, which is given by the area under which is covered by these yellow hashes, which is given by integral from 0 to w g of 0 dx. So, essentially, if we see that the area of this rectangle is given by g of 0, which is 1, and this width is w. So, the area of the rectangle is 1 into w because it is the length into the width, and we have integral of 0 to w g x dx. So, we can also write it as p hat is given by 1 by w integral of 0 to w g x dx. So, which is this equation, p hat is the area of the curve divided by the area of the rectangle is 1 by w integral from 0 to w g x dx. Now, here again we have a hat on top of g because this again is an estimate, we have not yet figured it out completely, but from here we know that once we have figured out an equation for g of x, we can compute p hat. So, even though p hat depends on a number of variables, now we are coming close to a way of mathematically getting to the value. So, when we perform this computation on a computer, we need to model it in some way. So, when we use a model of Gx, there are 4 criteria that we use for a good model of Gx. The first criteria is that of robustness. So, robustness means that the model should be able to fit a variety of possible shapes of Gx, which means that in certain situations we can have that we have a good detection at, so here we have distance versus the number of animals detected. Now, in certain situations we will have a situation in which we have a very good detection very close to the transect line and then practically 0 detection everywhere else. So, a situation would mean something like when we are walking on a patch of grassland and there is a very small species. So, if it comes on right on the transect line, we will be able to see it, giving a very good detection, and as soon as it moves into the grasses, we will miss it, giving a detection of nearly 0. Now, another situation could be where we have a detection which is roughly the same everywhere. Now, such a detection is possible, say when we are looking at elephants in a grassland. So, the elephant being a large species, we can see it even at a very great distance. So, its detection would be nearly equal everywhere. Now, when we are choosing a detection function, it should be such that it explains both of these extremes and anything else that also can come in between. So, that is the first criterion. Now, the second criterion is that of a shape criteria. Now, a shape criterion means that when we are saying that when an animal is right there on the transect line and we are able to see this, if the animal moves a bit from this transect line, do we see it or not? Now, in most situations, if you are able to see it right there on the transect line, even if it moves a bit, you will be able to see it, which means that when we are selecting any detection function g of x, then it should have a slight shoulder right near the transect line. So, a shoulder right near the transect line would mean that we will not be having a detection function that just moves like this, but we will prefer a detection function that has a slight shoulder here and then moves down. Now, this criterion, if we put it mathematically, we will say that the slope at 0, that is g prime x at x equal to 0 or also g prime of 0. So, it should be as close to 0 as possible, but it will be a finite value. So, that we have a small shoulder at that point. The third criterion is that of pooling robustness. Now, pooling robustness means that the model should work even when there are several unrecognized factors that are affecting detectability. So, as we had seen in the last lecture, there are a number of unrecognized factors and our model should be able to incorporate most of them. The fourth is estimated efficiency, which means that the results that we are getting should be precise, as precise as possible. So, when we write g of x in a general form, we can write it as g of x is given by a key function plus an adjustment function. What this means is that the key function refers to a shape that we have chosen for our g of x. So, if this is our shape for the g of x and if there are some points above and below, so the key function would give us the general shape of the g of x and adjustment functions would be used to modulate our curve, so that all these points that are coming out of the curve are also taken care of. Now, there are 4 key functions that we normally use. So, key function is telling us the general shape of g of x. So, the 4 functions that are commonly used are uniform function, half normal, hazard rate and negative exponential. Now, what do we mean by a uniform key function? It is very easy to remember a uniform key function by taking the example of elephants in a grassland. So, at very large distances from us we will be able to see the elephants. So, our detection probability is going to be the same everywhere. So, this is how uniform key function looks like. So, here we have that g at 0 is 1 and even at large distances g of x is 1. So, this is a uniform key function. The second key function is given by the half normal key function. Now, what is a half normal key function? Now, a normal curve is what we observe in most situations as a bell shaped curve. So, this is a bell shaped curve. Now, a bell shaped curve is the most common curve that is seen in biological samples. So, for instance, consider the heights of students in a class, or say the weights of different students in a class. So, there would be a midpoint at which most of the weights hover, and then if we go towards the extremes, then there would be less and less number of people that are having those weights. So, for instance, in a class where say the midpoint is 40 kgs and the extreme number of people that have the weights say 50 kg and 30 kg. So, we would find that most of the people have weights that are very close to 40 kg. So, which is so this is the number of students, and on the x axis we have the weights. So, we will find that most of the students hover like this in this region. So, they have weights somewhere say 40 kgs, 40.2 kgs, 39.5 kgs and so on. And then if we look at the extreme levels, so there could be a student that has a weight of 50 kg, there could be say a few students that have the weight of 30 kgs, but in general we will see a bell shaped distribution. Now, a half normal takes one half of this part. So, this is a half normal, this shape. So, if we take such a shape for our detection function, we have that at the top we have g at 0 is 1. We also have that g prime of 0 is 0, which means that we have a small shoulder here, and this is another curve that we can very easily use. Now, the equation of the curve is given as g x is e to the power minus x square by 2 sigma square when x is less than equal to w. Now, for this particular course it is not essential to remember all these formulae, but just a general shape would suffice. So, basically this is our half normal. So, you can have different kinds of half bell shaped curves depending on the values of. Now, the next function for g of x, the next key function is called the hazard rate function. Now, we can understand a hazard rate function by looking at its shape. So, a hazard rate function has a broader shoulder as compared to half normal, and then it goes down with distances x. What do we mean by this shape? It means that at g of 0 we have it as 1 and g prime of 0 is 0. So, we are having that at the top right on the transect line we have a detection probability that is 1, and also we have a shoulder here. But how do we understand this curve intuitively? Let us consider an animal that shows a phenomenon called flight distance. Now, what do we mean by flight distance? So, suppose you have an animal here and suppose this is your position. Now, if the animal has observed you and it freezes, so when we say freezing, it has observed you and just to avoid detection it just stands still there. So, it will not move. So, now as you go on approaching this animal, there would be, so you came to say this point. So, this is your point P0, then you reached a point called P1. Even at P1 this animal is just standing there, it has just frozen at its place. So, you go a bit closer at distance P2, at point P2. So, even then it is freezing, but then as you go on increasing your nearness to the animal, there would be a certain point say Px at which this animal now thinks that you have observed this animal and you are coming to grab this animal. So, what it will do is it will run at that point. So, this distance of d till the point where the animal is able to tolerate you is known as the flight distance. So, if you are anywhere greater than the flight distance, the animal is going to freeze. If you are anywhere within this flight distance, the animal is going to run away. Now, how it affects our detection of the animal is that when the animal is standing frozen right there on the ground, when it is not moving and it is very well camouflaged, so we will not be able to detect that animal. But when this when we have approached this animal and when we have crossed its threshold of the flight distance, this animal will run away. Now, when an animal moves, it is very easy to detect that animal. Now, coming back to our curve, it shows that when we are at a distance of d from the animal, this animal runs away, which facilitates its detection. So, we have this broad shoulder on the top. So, till our distance d, we have a very good detection and nearly a complete detection. So, we have g of x is equal to 1 till our distance from the animal is less than or equal to d. But right after that, as soon as we have crossed this distance, so now the animal is comfortable. Now, this animal is not going to run away. So, we will not be able to detect this animal. So, when you are walking on a transect line, if the animal is at a certain distance and if this distance is greater than the flight distance, then the animal will just stay frozen there and it will not move. So, we will not be able to detect this animal. But as soon as we have a situation when this animal is closer to the transect line than the flight distance. So, basically what we are saying here is that when we are moving on our transect line A to B, here we have this animal and we are at this position. So, at this position, this distance is greater than d. So, this animal is just standing there. But then when we came to this point, now our distance from the animal is equal to d. So, even here the animal is right on the threshold, but we take one more step on our transect and our distance becomes less than d and this animal flees away. So, essentially we will be able to detect that animal till a distance of d, which is shown by our curve. So, this is our general shape of the hazard rate key function. So, we have a shoulder on the top and then this curve moves down. Now, it depends on two parameters. So, the parameter sigma regulates the slope of the curve when sigma varies at a constant of beta. So, this is how it will look. So, we have kept beta constant at 1 and we are varying sigma. So, this is how the shapes are varying. And similarly, so again for this course it is not essential to remember the formulae, but just a general shape of the curve in your minds will suffice. Now, the next function, the next gx is given by the negative exponential key function, which has this formula of g of x is exponential of minus of x by sigma. Again, looking at a common shape, we have that at this point we are able to detect the animal, but right after it the probability goes very close to 0. Now, it is important to note here that in the case of a negative exponential key function, it does not have a shoulder on the top. So, essentially our criteria of g prime of x at x is equal to 0 should be 0 is not fulfilled by this negative exponential key function, but it also becomes essential for those situations in which you are able to detect your animal only when it is right there on the transect line. So, this happens in situations when we are considering say reptiles. So, if there is a snake, and snakes are generally highly camouflaged animals because they are predators. So, if there is a snake that is right there on your transect line, you will be able to see it, but as soon as it goes into the grasses, there is very little chance that you will be able to see it. So, this curve essentially shows that phenomenon. So, right there on the transect line you have a good detection probability, but then it drops down very fast. So, these were the key functions. Next we have the adjustment functions. So, we have the g of x is given by a key function plus an adjustment function. So, your adjustment function can also be written as a key function multiplied by something. So, the adjustment functions are generally used as in two ways. So, two commonly deployed adjustment functions are the cosine function. Again, the formula is not important, but the shape is important. So, in the case of a cosine adjustment function, we will have these wavy lines which represent our cos theta values, and the other one is a simple polynomial adjustment function. So, this is our simple polynomial adjustment function. So, basically it could be something like y is equal to x, y is equal to x square, y is equal to x cube, or any combination of these. So, essentially what we have said is that the g of x is a key function plus an adjustment function. Now, key function is the basic shape of g of x, and there are four basic shapes that we have seen. One is uniform, which goes like a straight line. The second is a half normal, which is half of our bell shaped curve. The third is a hazard rate, which essentially is a shoulder followed by going back to 0, and the fourth is a negative exponential, which is just going to 0 right after the value of 0. So, here we have, in all these curves we have the g of x versus the x. So, key function is explaining the basic shape of the curve. Adjustment function adjusts the key function to include points that are not exactly on the basic shape. And here we saw two adjustment functions. One is a cosine function, which moves like this, and the second is a polynomial, which could be x to the power alpha 1 x to the power 0 plus alpha 2 x plus alpha 3 x square plus so on. So, essentially we are saying that our function here is either y is equal to x or x square or x cube and so on. So, basically these are the two key things to remember: key function and adjustment function. Now, how many adjustment functions do we use? So, here we have this concept of bias and variance. So, essentially if we consider the number of parameters, so if there are more number of parameters, what happens? Now, bias is the difference between the readings and the actual reading. So, if we remember our case of the bull's eye shooter, so this is our bull's eye and the shooter had all the points here. So, this distance is our bias B. Now, if we are including more and more terms or more and more parameters into our equations, the bias reduces. Why does the bias reduce? Because in place of having our, so this was our model of g of x, but actually our points were lying somewhere here. So, we included some adjustment functions so that all of these points are also covered. So, in that case the level of bias reduces, but at the same time if you look at variance, the variance increases, because what we are doing in this case is that all these points that we were having, so these points that we were considering, these points might not be representing the actual signal, but might actually be representing the noise values. So, noise values means that these are those data points that we are not very sure of. So, in that case, as we increase the number of parameters, the amount of variance goes on increasing. So, the next question is, how many parameters should we take? It is always best to take those number of parameters for which your bias is less, but at the same time your variance is also less. So, essentially a mid number of parameters. So, do not over adjust your g of x, do not under adjust your g of x, take a middle point. So, we should target a middle number of parameters. Now, another thing is truncation. Now, in the case of truncation, what we have is that when we consider the value of g of x versus x, we can also say that for these values we are very much sure that of having an actual observation. So, essentially when you are moving on a transit line and there are animals that were very close to your transit line. So, you are very sure that you have seen this animal, but then consider an animal that was very far away. So, now you are not sure whether what you saw was actually an animal or whether it was, say, a rock of the same color. So, essentially at very large distances, was it an elephant or was it a black colored rock? You are not very sure. So, in those cases, to improve our accuracy, we can just say that we are going to disregard all the values that we got after a value of x is equal to x0. So, this value of x0 can be chosen by us, and we can say that any values that were greater than this distance, we are going to truncate it out. So, truncate it out is, it means that we are going to leave it out in our calculations. So, we typically truncate around 5 to 10% of the observations. So, those observations that are very far away from the transit line, in order to improve the fit of the model without resorting to a very large number of parameters. Now, once we have selected a model and once we have tried to fit our model, so essentially when we have some points, we could fit a model, say like this, a negative exponential model, or we could say that we are going to fit a hazard rate model, something like this, or we could say that we are going to fit a half normal model, something like this. Now, you can see that none of these models is actually catering to all the points that are there on our data. So, how do we assess which model to take? So, essentially there are three easy ways of assessing a model's performance, which go by the name of likelihood, AIC, which is Akaike information criterion, and absolute measures of model fit. So, for the purpose of this course, it is not essential to get into the details, but basically we should only understand that it is mathematically possible to understand which of these different key functions and different adjustment functions is mathematically the function which is using the least number of parameters and is able to explain our data points to the highest degree, which is actually what we want. What we are looking after is just a value of g of x from which we can get a value of p hat, and we can have different g of x's as we can see on the screen. So, we can have different g x's. So, we can have a negative exponential, we can have a half normal, we can have a hazard rate and so on, but which of these should be taken, we can compute it mathematically. So, our computer can give us the results. Another concept is that of the effective strip width. So, once we have decided our g of x, the next thing that we want to compute the density is, the density is given by the number of animals that were there divided by the area, and the area is given by the length of the transit multiplied by twice of the half width. So, we can also multiply it by k, which is the number of transits that we took, and the n can also be modified as n by p hat. So, we can compute this p hat with our g of x by 1 by w integral of the g x, but and we know the number of transits that we have walked, we know the lengths that we have walked. Next thing to figure out is w. So, can we have a mathematical function through which we can find out the w? So, here comes the concept of the effective strip width. The effective strip width mu is defined as the distance at which as many objects seen beyond mu are missed within mu. So, what we mean here is that in the case of our curve of g of x, we can always draw a line. So, at this distance w, so anything that is below our curve of g of x is what we have detected. So, this portion which lies within the value of g0, which is 1, and g of x is the number of animals that we have missed out at distance w. On the other hand, the number of animals that are seen beyond this width of w is given by the area of the curve below g of x, so which is given by the blue shade here. So, at every distance w, there will be some animals that have been missed out within this distance, and there will be some animals that were seen outside of this distance. Now, as we go on increasing this w, we will reach a point at which the number of animals that were missed within this distance given by the red color is the same as the number of animals that were seen beyond this distance. So, both the curves have an equal area. So, at this distance we say that the width is given by mu. So, mu is the effective strip width. So, why do we need mu here? Because coming back to the screen, we require a value of w, and when we use this value of mu as w, we will get our results. So, essentially for n observed animals, the effective strip area is given by twice of mu, so which is twice of the half width multiplied by the length. Now, here this capital L, because it is small l into k. So, this is the effort or the total distance that we have moved. So, the density of animals will be given by d hat is equal to n divided by this effective strip width 2 mu of L, and the number of animals will be given by this d hat into A. So, it is n into capital A divided by twice of mu hat into L. Now, all these formulae are not important for this course, but what we need to remember here is that it is possible to find out a value of g of x from which we can get a value of p hat, and it is possible to compute your mu hat or the effective strip width also from the g hat, from the gx curve. So, once we have done that, we can use this value of mu into our equation to get the density figures and the estimates of the animals within our forest. So, in these two lectures on distance sampling, we saw how we modulate our equations for the density. So, density essentially means number of animals per unit area. Now, we saw that we are missing out some animals, so we included the concept of p hat. So, we modulated our value of n by p hat to incorporate those animals that we missed out. Now, we miss out the animals because of a number of factors. So, it could be anything and everything that can influence our detection of the animals. Now, to compute a value of p hat, we went through this exercise of finding out a curve g of x which had some criteria, that g at 0 is 1 and there is a small shoulder at distance x is equal to 0, and from that we can compute our value of p hat, or similarly we can compute a value of mu hat. So, once we have done that, we get our values of n and d, which is the estimate of the number of animals in the forest or an estimate of the density of animals in the forest. So, distance sampling is one very important technique that we are using these days to estimate the number of animals by incorporating all the factors or most of the factors that can influence our detection of the animals. So, that is all for today. Thank you for your attention. Jai Hind.